But what would it take for life to all be connected? I think this is an important question because the science that's been done because of this question actually reveals the kind of life that we have on planet Earth. It reveals the complexity in the systems and structures and information within life. That's what we're asking next. What does it take to make a new animal? Now, scientists have identified 3 components needed for new body plans. First, new functional protein folds. Now, a protein fold is a combination of about 300 amino acids bonded just right. You might think of it kind of like origami. They're folded just right, making a particular function possible. Now, what are the odds of getting it bonded just right in order to make any function possible? Well, it's like a little molecular machine. In fact, it's been calculated that we only have one chance in 100,000 trillion. It's not just a random glob of goo. It's bonded just right, like a little molecular machine. Now, what's that look like? Molecular biologist Douglas Axe calculated that the probability of generating just one new functional protein fold from an already existent functional protein fold, by the way, that's a generous assumption that one already exists, were the odds of getting a new one from that pre-existing one. He said that if all organisms to have ever lived on planet Earth generated one new sequence, tried one new fold, only 1/10 trillionth of sequences would have been tested. And that's just for one new functional protein fold. The fact is that we actually need whole new systems arising together for a new functional body plan. There's no hope in the history of life on planet Earth to get just one, but how much more whole systems? And this is exactly what we have to assume happened repeatedly in the history of life on planet Earth to get vastly new body plans with new functions. So new functional protein folds as a new component seems unlikely. Second, new genetic information. This is the second component that is needed for a new body plan. Now, to unpack this, let's explain what we mean by genetic information. Genetic information holds what theorists call specified complexity. Let's unpack that term, what that means. First, let's start with complexity. A simple sequence is subject to redundancy and patterns. Now, it's actually that redundancy and patterns that keeps it from being able to hold higher complex information. So a complex sequence, on the other hand, is not subject to that redundancy and patterns, making something higher and more significant possible. But that doesn't mean that's what you're going to find. That's where the specified part comes in, because you see, unspecified complexity, or what you might call mere complexity, it has no particular arrangement, no particular parts, and there's not going to be a particular function. In other words, it's just kind of gibberish. But the specified part says that you have the specific arrangement of specific parts resulting in specific function. This is kind of what we find in an English sentence. In the sentence, you see a word like his. It's not just a random collection of things. In order for there to be the function, you need the specific collection of the specific letters in the specific order and placement. That's specified complexity. And that's exactly what we find in DNA. DNA has genetic letters arranged just right together, making the function possible. Now the question is, can you get new genetic information, new specified complexity in the genetic code from mutation? Now mutation has a couple of ways that it can happen. You have inversions, you can have insertions, you can have deletions, a couple different tools. But just for the sake of simplicity, let's just say, in this sentence, what happens when you just start randomly changing things? Just one letter at a time. Are you getting new information, new function, a new meaning, a new sentence? No, you see randomness corrupts order. The function was lost. And that's what we find, that information is lost when you try to corrupt the specificity. Specified complexity, the specificity is crucial for the function. Now, randomness is the opposite of specificity. So when you start randomly changing specificity, you don't get new specificity, you get less specificity, you get less information. We see that random changes don't increase order and add specificity and information, but rather they corrupt order and destroy specificity. Now, what does that even look like in the context of life? We see that degraded order results in degraded function. When we start corrupting it, it's going to impact the function on the other end. So you're going to have degraded function. Now, when you have degraded function, you have a loss of fitness within the organism. Now, if you know something about natural selection, survival of the fittest, if you have a loss in fitness, you're not going to go on and propagate your genes and create a new whole type of organism. You're actually going to die out. It's going to cut it off there. So ironically, natural selection actually puts a limit on the amount of mutation that can happen within the genetic code. As explained by Stephen C. Meyer, since genes, like English sentences, contain sequence-specific functional information, multiple changes in the genetic text will inevitably degrade function or fitness long before a new functional sequence will arise, just as random changes in a meaningful English sentence will typically destroy meaning long before such changes produce a significantly different meaningful sentence. You might imagine that after maybe a lot of changes, maybe you get some amount of small regions of order as a result of random change. But that's exactly what time doesn't allow you, because as soon as you start losing order, life is going to end there. You don't have time to just keep on messing with it, because life needs that function in order to survive. So new genetic information is also doubtful as a result of mutation. But the last component is new epigenetic information. Now, epi just means beyond, so epigenetic information is information beyond the genes. A couple examples include cytoskeletal arrays, microtubules, centrosome, membrane patterns, membrane targets, ion channels, and the sugar code, just to name a few. Now, what do those do? What does that mean? Do we have any Lego fans in here? Yeah, I know there's a big push for Legos right now. I did Legos as a kid, but then, I don't know, they're so expensive, you know? It's plastic. I don't get it. What's with that? But you might think of new epigenetic information as kind of like a Lego instruction manual. It tells you how to put the pieces together into a higher complex structure. For example, take one of these boards. Just having the pieces, does that tell you how to put it together into the higher complex structure accomplishing a function? No, you actually need information beyond the circuit board in order to put it together. Or take a house, for example. You need more than just the raw materials. You need more than just pavement and glass and concrete in order to build a house. Instead, you need more information. You need a blueprint in order to put it together into this higher complex structure. That's what epigenetics do. They collect proteins into cell types into tissues, tissues into organs, and organs and tissues into body plans. Now the question then is, can we get new epigenetic information via mutation? Now it's doubtful for two reasons. First, these structures are really too large to be subject to the general sources of mutation, such as radiation or chemicals. Second, if you were to impact them in any significant way, which the only time you can do that is during the early embryonic phase, it would actually be fatal to the organism and its development. So for this reason, it's very doubtful to get new epigenetic information. Now those are the three components that scientists have identified new body forms require. We saw that the odds of just getting one by itself is incredibly improbable. How much more all three together? You need all three arising together, making the new body plan. Just one or even two without the third doesn't give you a new functional body plan. You need all three. So that poses a problem. As explained by biochemist Paul Nelson, a biologist, if the only kind of mutations that can conceivably produce enough morphological change to alter whole body plans never causes beneficial inheritable changes, then it is difficult to see how mutation and selection could ever produce new body plans in the first place. Thus, life seems to be isolated within major kinds. And that's what we've learned from contemporary science, molecular biology, genetics, and epigenetics.